first team is team 11.
Yeah.
Could use a 10-minute timer because I guess it's supposed to be very hard to timer sometimes.
Well, we'll try our best.
Okay. I think we need to go through them pretty
All right.
quickly to the time it says. So I think we can start as soon as you're ready. Okay, you ready?
Yep.
Okay, we're gonna start the presentation. Go ahead.
Hello everyone, this is Team 11 and my name is Howard. Today we're going to present on our project BioRag, which supervised learning retrieval augmented generation in biomedical QA.
There's a microphone.
Oh,
Which one's? I'm not sure. I can give you like an extra minute if there's a microphone.
hello? Okay,
I think so.
a little bit. Okay, so before we dive into our topic, I want to introduce what retrieval augmented generation does. In short for RAG, it allows large language models to reference a knowledge base outside of its training data sources before generating a response. In short, it uses external database to enhance large language models' text retrieval capabilities. So why do we want to use RAG? That's because currently the large language models have limitations on giving precise answers, especially for scientific fields. And for people who are familiar with the large language models, you have heard the word hallucination. That is, the model tends to be confident about what it gives, but maybe it's just wrong. So with RAG, we can also eliminate these hallucination because we have a concise database. Also, we could reduce the computational expense because we can limit, shrink it down to a certain size. What? Yeah. And so what we want to do with our project is we want to grant easier access for non-professionals to retrieve biodata. So as we all know, the natural language query is much easier than machine language query. Like when you ask something to ChatGPT, it's much easier to get an answer than for non-professionals to learn some language like SQL to query a database. And so what our work is inspired by a previous work called Knowledge Graph RAG in natural language processing. And we applied to more biomedical, bioinformatics data sets to allow it. Yeah, and for our method to work, there are two phases. The first phase is setup phase. We need to build a vector database with the bio data set that we have using something called ChromaDB. It's a package from LangChain. And for the second phase, with the inference phase, I want to show you a demo with our program. Yeah. So as you can see, this is our UI demo, and there are 5 steps in querying a RAG. First, we need to enter a question. For our demo, we use the DustCov2222 binds to SARS-CoV WT. That's an antibody and with an antigen. So first step, we will extract the entity using a large language model. Here we use GPT 3.5. And it will see that the prompt was mainly about the antibody there. And then in the next step, it will match the extracted entity to the spoke. It's a database of databases. It's actually a knowledge graph. And we can use this like a lookup table. We use the extracted entity to query this database. And then it will retrieve a context related to such entity. Afterwards, we'll use a embedding model to prune this extracted context. And then we will have a concise answer. Afterwards, it will query the large language model again with our question and the extracted context. Now, let's further move on to pass it to my.
Hello everyone, my name is Qinglong, and this is a naive RAG workflow overview. So this workflow can separate in 5 modules. So first we have our knowledge base and retrieve module and the prompt module, large language model as generator, and the user interface chat in a chat box. Yeah. So what we do is we substitute the flattened structure knowledge base to knowledge graph. Is a hierarchical structure data set, and we adopt a dual level retrieval algorithm to substitute the naive one, and we tried 5 different strategy in prompt section. So here we adopt the dual level retrieval paradigm, which is we extract from local query keywords and global query keywords, use by prompting the large language model and doing similarity matching to the context in the knowledge graph, and it will generate two set of candidate entities from 2 levels, and gathering the one hop subgraph from with the additional layer of context, and then this will combine and to the final retrieve context, give it to the language model to generate the final answers. And here is our strategy, prompt enrichment generation. We tried 4 different strategy to improve the performance on our benchmark. So first, mode 1 is to convert the retrieval content in the JSON format. Mode 2 is we add a prior knowledge to enrichment the prompt. Mode 3 is a hybrid mode to combine mode 1 and 2, and mode 4 is we proposed it's something like chain of thought, but it's used in the knowledge base scenario, the knowledge graph scenario. And
I will quickly talk about our experiments and results. So first we have 2 benchmarks. The first one is multiple choice questions, and each question has 5 options with only a single correct answer. The second one is the binary questions. So if we impose the chroma as a answer, we expect our framework to output true. Due to time limit, we haven't run this one. And for our experiment settings, we have 2 baselines and 4 adjusted frameworks and 1 ablation study. And we use Gemini to test the MCQ benchmark and GPT 3.5 for generation demo in the previous slides. And notice that we embed our disease concepts with a sentence transformer called or mini LM L6 V2. And this is the table of our results. So for the first one, the baseline one is the framework with the additional context information from the knowledge graph. The second one is with the KGRAG. And for the mode 5, it's just a ablation study compared to mode 4, which adopts a 2-stage strategy like you with an additional prompt that will allow the large language model to make a proper option, and it has the highest accuracy. As you can see, incorporating hierarchy structure, adopting RAG, reformatting the retrieval context, and all these ways can significantly improve the accuracy of our biomedical LM tests. And in the future, we want to adopt multimodal RAG framework and run the second benchmark. And for now, our tests are in static knowledge scope in the spoke database. We want to apply our frameworks in a more dynamic knowledge scope. And that is all for our project. Thank you for listening.
Hello everyone, we are team 17 and I'm Tianyu and this is my collaborator Yurei. So our topic is about the drug discovery through this curve-based molecule optimization method. And we adopt this architecture from a previous paper and we changed our target to something else. Like we use 2 examples to show this method on other possible drug discovery background. Yeah. So this is our outline of the talk. I'm gonna talk about the motivation, background, and method. And Yuri is gonna share where we acquire the data and the results of primitive results currently. Yeah. So why we want to do this project? Because we want something to help to optimize the molecule structures to certain drug method, because we want to change the molecule structures to certain targets to optimize its structure, like something like that. Yeah, and this method so basically this method is we adopt from this paper Hoffman in 2022, and basically what they did is just that they import the SMILE sequence. It is a 1D string to fully reflect the 3D molecule structures of the small chemical molecule structures, and we import that to an encoder, and it will go to a latent space. And also we want to, latent space will get something like a continuous place with a vector, and we use that vector to do some property evaluation and to get some and to get a loss function from there and to train the iteration until it satisfies some criteria. And finally, our decoder is just to decode the latence vector to the SMILE sequence, which can be further converted to the chemical structures. So we choose these 2 data sets mainly because it's dopamine. DRD2 is a target from previous research. It's a target from the antipsychotic drugs and for the Parkinson drugs, some neurological stuff. And so is the GSK3 beta, and GSK3 is also some potential target for the Alzheimer's disease. And this is just a PDB database. I acquired this structure from their database, and yeah, that's it. So this is the entire workflow of this method. So as I said, like you get some chemical molecule structures, and you first to use something to convert to a SMILE 1D sequence. That's the input for the encoder. And you put it into encoder, you get some possible embeddings, which is Z. And and you evaluate some properties on this in the latent space. And this is some property you are going to give it like a score function. So in their original paper, they measure something like a drug similarity or something like a possibility to synthesize this molecule in a chemical workflows. Yeah, so something like that. But we change it to the score like DRD2, which is a score for like what's the possibility this chemical molecule would bind to that dopamine receptor. And the other 2 score, like I said, where is that? Oh yeah, this here. So these are two scores for the JK3 beta, and JK3 is the possibility this chemical molecule can inhibit this protein or receptor that can get some positive results from there. Because if we inhibit their function, it will somehow improve their neurologic disease. Yeah. So this is the mathematical part. So basically, we have a pre-trained encoder and a decoder. The encoder is just to convert the SMILE sequence to like a latent space vector. And in this place, it is just like hidden space. And then, since it is the input here is the SMILE sequence, it is not a continuous one. So we cannot use a continued gradient descendant way, normal way to do that. So instead, this method uses sampling Q samples with one step length from this nearby region, and randomly, and it can get Q samples. It's the number of the samples, and it will try to verify if it's a valid SMILE sequence before it gave out that place. And based on that, we will calculate the loss function, and that's the way they do the gradient descendant. And if you put a score that has a requirement, a threshold, it will check if they satisfy that threshold. If it satisfies, it will just leave. Otherwise, currently, we just go this for loop for like t times. Our t is like 1,000 iteration at this moment. After that, you will update it to your final data set. That's it.
So for our experiment, we generally use two data sets. Like the first data set is called zinc. So it's like a large-scale data set that often used for like ligand discovery or drug discovery problems. And so we initially used that for the DRD2 and GSK3 beta optimization experiments. And because we want to select those molecules that are more potential to show that inhibit to these two. And so we calculate these two score for all of the molecule in this data set, and we select the top about 100 ones for these two initial experiments. And another data set we use is actually more targeting on like the GSK3 betas inhibition. So they have some like small data sets that are we think that may more suitable for the problem we are like researching on. And we use this data set for the optimizing of the GSK3 beta and JNK3 inhibition together. And our result on the DRD2, so from the loss curve on the left side, so the loss curve here is like the DRD2 like minus value. So it seems that through the optimization, it is going down. And we also show like the original molecule graph and the molecule graph after the optimization. And so we also do a graph on the whole, like the top 100 molecule we are doing experiments on. So it seems that for the mean improvement, it has about 0.3, and the standard deviation for improvement is like 0.2. So for almost all the like molecule, they have an improvement on the DRD2 value. And for the GSK3b, so for this like molecule we are selecting, so it seems that it has a good job in the loss and have some very minor change in the like its graph structure. But from the data set, like the overview result, it seems that it's not as good as the DRD2 problem before. So, and that's kind of the reason why we want to change the data set to something more targeting on the GSK3b, that the molecule in it have more potential to become like inhibit to the GSK3 beta. And further do this kind of combinational optimization things on our new data set, but currently we don't have, like, we don't do experiment on all of the molecule in the data set. So from just this molecule we are showing, it seems that it do have improvement on both of these two properties. So in the future work, we are planning to like do more on experiment on this combination optimization problem, and we're also thinking about like do more data filtering and switch data sets to other like more targeting data set on this kind of problem. And this is our reference, and that's all. Thank you.
Team 6.
We're going to use the mic.
Heck yeah.
All right. So, you ready? Big. Slideshow? It's over here on the bottom. All right. So this is our final presentation. This is our final presentation, and we.
Well, you can use the microphone.
Okay.
These are all been recorded.
Fair enough. Right. So we're Team 6. I'm Akash. This is James. That's Matt. And our final presentation is essentially we're trying to adopt a language modeling approach to trying to design DNA binding proteins. Can you go to the next slide, please? So DNA binding proteins are rather important in the body. So they possess several important functions, including regulating gene expression, enabling gene editing, CRISPR, for example, through CRISPR-Cas proteins, changing gene topology, and more. And so developing de novo DNA binders could potentially help us target on and off regulate specific genes and lead to more impact in like healthcare, agriculture, or other fields. So our motivation for this problem for adopting a language modeling approach essentially is that designing DNA binding proteins, like DNA binding proteins themselves are like it's like a fundamentally structure-based issue. Where the DNA needs to align along with the major groove, which is, if you imagine the DNA is like, it's a helix, and there's one side that's more exposed than the other. So the major groove is the more exposed side. So the proteins need to sort of recognize the nucleotides and the base pairs on the more exposed side and sit correctly within that groove, and also wrap around to the other to make sure that the protein stays in place so that you're able to do what the protein's supposed to do. So as such, like previous approaches to designing DNA binders have been heavily structure guided, such as the listed paper from Glasscock, which is like from David Baker's group, which basically what they did was tried designing particular scaffolds that would sit well around the major groove, a very specific kind of scaffold called a HTH scaffold, and which was basically like like a forearm essentially that's going to sit around the DNA. And there has been some interest in, actually significant interest in language modeling approaches to designing DNA binding proteins. Like we saw in the EVO paper that they tried designing Cas proteins. And there's also a company called Profluent, which has raised, I think, like $35 million for designing CRISPR proteins using LLMs, essentially. Can you go to the next slide? So for this project, we essentially wanted to explore if we could adopt a purely language modeling or sequence modeling approach to this problem without incorporating any structural biases into our data, which is a departure from previous models where we've also seen the incorporation of structure information through the sequence itself, such as like, for example, how we saw in fold-seq. So this is what we're doing, and I will let Matt take over so that you can talk about this. Sorry, James, take over so you can talk about this problem definition.
So, as we have defined on our problem, the first question is, can we define de novo DNA binders using only a sequence modeling and language modeling as approach without using any sort of a scaffolding or structural information whatsoever? And So we have a few criteria for what we want to do. Like we want to first of all generate like novel proteins with a low template modeling score, which means that like the proteins would preserve like the motifs of known DNA binding modes. And the second objective is that we essentially want our proteins to work. So we want them to be able to bind to the DNA sequences that we want them to bind to.
Okay. And so that's the problem definition. Now I'm talking about like how we actually do that. You could split that problem down to two different steps. One of them is generating DNA binders, and the other step is to seeing if the protein actually binds to the DNA. So I'll go over the first part because that's the part of the project I worked on. Slide. So to make DNA binders, first, we want to actually like have some DNA binders to work off of. So I collected data from UniProt. Initially, we got all DNA binding proteins, but when we tried working with that, then the model just did not work as well. So we thought that if we were to switch to mammalian proteins, then there'd be less diversity with the proteins, and we'd get better results. So in total for our data set, we have about 8,000 proteins. Slide. So the next step to this is that we fine-tune ESM, because right now ESM was trained on all proteins. So we wanted to generate DNA binding proteins. So we did mass language modeling on the DNA binders in our data set. And here's some stats about the fine-tuning. So we used a 650 million parameter model. The bigger model just like crashed NCSA, so I could not use that. Training time was about 5 epochs. We stopped once we saw that the test loss plateaued. Once the test loss plateaus, then the model is probably overfitting. And here's some other statistics. The train loss went from around 1.9 to 1.1. Test loss from 1.9 to 1.2. We did a 80-20 test split. So we know that the ESM is actually learning on the data. Slide. Okay, and the next step after we have a pre-trained, or after we like fine-tune ESM, now we're going to sample from ESM. This is very similar to what we did on a homework problem too. We use Gibbs sampling. So initially we started with a protein sequence that was just mask. And then at every iteration, we would change an amino acid from the logits that ESM2 outputted. So here is like one protein sequence. I cut it off just for brevity. But at every line is a new iteration. So you see at some points let me see. An S turns to an M, for example. And in order to generate the proteins, we found that if we started with a higher temperature and then exponentially lower the temperature as iterations increase, we get a high variety of proteins. We generate proteins from various sizes from 128 to about 350 amino acids, just so we can capture more of the protein binding space. And we realized that we had to do a lot of iterations to actually generate the proteins that work. And then I'll give it to James now.
To measure or like generate proteins, we have to use like multiple sequence alignments to other proteins. For example, how similar are the generated proteins to each other, and how similar are the generated proteins to other proteins within or out of the DNA binding domain. So this gives rise to a field called homology analysis, and this is based off of kind of an assumption that that sequence homology is related with structural or and functional homology as well. And so with each a novel sequence, we can perform something like a center star alignment as well on the on a de novo a protein sequence.
Sure, we operated in the sequence space, but ultimately this is a structure problem, and what the proteins that we design need to sit well with the structure. So for validation, we essentially went for a structure validation approach. So we used AlphaFold 2 to sort of visually evaluate the structure, aka eyeball, and see if it actually looks like what a DNA binding protein would look like if there are binding modes that are preserved. Wait, do the videos not play?
The videos are on your Google Drive, so I don't know if this kind of access to it.
Oh no, this is, wait, can we?
If you change the access.
No, I'll just show the videos in giant form. I I think they're probably the coolest part about it.
Yeah.
But anyway, so this was one of the proteins we generated. This is our best looking protein. All our other results look rather sorry, it's like expectations is a reality meme. So this was the one that visually seems closest to, sort of trying to even preserve a HTH motif, because you see that there's one sort of forcep that's an alpha helix, and then a linkage over there that sort of bends over to the other place. And then we have other results. I'll also show you the more sorry ones.
Can I also add that we've like, just like in the actual paper, we found that like the longer the protein sequence, the more likely it is to just like look like this.
Yeah, look like noodles.
Yeah.
We also have other results. I'm afraid. Oh, you can see the PLDDT scores and the PDM scores. So our fundamental approach to sort of using PLDDT and PTM over here, where that PLDDT sort of guarantees local stability, that a protein in the local neighborhood looks like what it's supposed to look like. And PTM low enough is probably an indication that it is either A, the model is completely unsure about the global structure, or if the protein seems to have some semblance of a defined structure, then a low PTM probably indicates that it's novel. But except for that the first protein that I showed you, I think the case for all the other results that we have is that the model is just unsure about what the structure is, which you also see over here. This does not look like a DNA binder at all from like any angle. That was one problem. Anyways, can you go to the next slide? Yeah. The results. No, the one after. The other results.
Also, should be wrapping up.
Yes, yeah, this is the last slide. So we have interpretation plus future results. Essentially, so we currently fine-tune on all mammalian DNA binders, but I think for future approach, like for a future direction, like by the time we submit the final report, we should probably restrict ourselves down to a single species, as we saw in, say, other papers like the EVO paper, which restricted itself to prokaryotic genomes and like a particular subspecies, I believe. And and our designing is currently unguided. So which means, like, as Matt mentioned earlier when in his part of the presentation, we're simply demasking. But we also have 2 planned experiment runs with like sort of targeted generation, where there's also a notion of what DNA segment you want to bind to. But yeah, is there another slide? I don't think there is. Yeah, references and questions.
Do we have the? Oh, okay. Yeah, cool. That's good. Hey everyone, we're team 3, and today we want to share with you our presentation. It's going to be on using convolutional neural networks to detect breast tumor malignancy. Which one? This one? Okay, cool. So our goal here is to explore and improve the current methodologies that we have to classify breast tumors, whether they are benign or malignant. So why is this topic important? It's important because breast cancer is it is the most common cancer for women in the US, according to the American Cancer Society. And it accounts for around 30% of all new female cancers, and it affects around 30,000 people per year. So the tricky part about this is not actually to find the cancer. It's actually to tell if the tumor is benign or malignant. Because benign tumors, well, they are non-cancerous, they don't spread, and life expectancy is not affected. But for the malignant case, depending on which stage you're on, your 5-year survival rate could be varying from 90% to down to 30%. So where does machine learning come in here? Well, the current workflow is we take like an X-ray and we first take a look at the area, and if we want to confirm if it's of the type of the cancer, we do like a autopsy, no, a biopsy. And machine learning could help with in between the X-ray and the biopsy, so that we don't have to perform as many intrusive experiments. But then we could also have a good grasp of what the situation we're dealing with right now.
Yep, so to briefly cover related work, I just want to focus on something you mentioned earlier. The highlight of our research is actually the feature selection. So getting a high accuracy with regards to results with classifying benign or malignant tumors doesn't always relate to having the best results, because there are a host of factors that could influence the results. So in terms of related work and what actually influenced us to go through this direction, the first paper we looked at was Spanhole et al., which demonstrated the effectiveness of CNNs on the Wisconsin breast cancer dataset, which is the one that we employed in this study. And they first achieved an accuracy of 98.6%, which is a really high benchmark. And then their key model features include transforming the numerical features into a 2D representation, which allows it to be used on a neural network. And then later on, there was an improvement by Nahid et al., which used CNNs in combination with long short-term memory, and they achieved a really high accuracy of 99.68%. And like, as I mentioned earlier, like, these are just numbers with accuracy, but there are still a host of factors that could influence these decisions. So getting a holistic picture and improving the model features is increasingly important.
So the dataset we used for this project is the breast cancer Wisconsin dataset. It's basically a bunch of numerical features that describes the different aspects of breast tumor, and it's like their shape and the textures. And for each of these samples, they are labeled as either benign or malignant. And before training our module, we conducted several pre-processing steps to ensure that our data was ready. So we applied standard scaling to normalize feature values. This step helps the module converge more smoothly during the training. And we also split the data into training set and test set as a 80-20 ratio. So we had a fair way to measure how well the module would perform on new data.
So as Arun mentioned, mainly we use literature-based selection for the model features. And so the features that we mainly focus on are things about the morphology, and which is like the texture and the shape features. So these include the radius or the texture, the perimeter. Perimeter is like around the nucleus, and the area compactness is like how big the nucleus is compared to the whole size of the cell, and basically the shape, symmetry, and concavity of the tumor. And this could tell us, like, if the tumor was more malignant, it usually would have a higher level of asymmetry. It would be more concaved, and like overall the texture would be more rough. So this was like for feature selection. And then for the hyperparameters, we utilize what we learned in class about like the learning rate, the L2 regularization. So learning rate helps us take steps, but we have to control it in like a step size such that we don't get stuck in a local minima if the learning rate's too small, and we also don't overshoot. So we actually do find like a minimum to drop into. And then regularization and early dropout helps us with overfitting, especially with like a smaller data set size. We want to make sure that our model is still generalizable. And also for the batch size, we are looking at like how fast this model updates per batch, and we also wanted to incorporate this into the training features.
So for module architecture, we designed a 1D CNN to classify the tumors. So we started with 7 chosen features, including radius and texture. So these features are fed into multiple convolutional layers, and each layer increases the complexity, and eventually extracts the pattern that might indicate malignancy. And to further stabilize training, we integrated a residual connection inspired by ResNet architectures. This helps the module preserve important information from earlier layers and make it easier for module to learn complex relationships. And after convolution and pooling steps, we flattened extracted features and pass them through fully connected layers and gradually narrow down to a single output, and that gives us the probability of the tumor being malignant.
Cool. And in terms of the methods that we used, we followed an iterative approach in building out the ML model. So we added more layers and features until we reached a target accuracy. For us, as we defined in our proposal, our target accuracy was an F1 score of 0.7, which we did achieve. Our baseline model, which we first started at, was a 2-layer convolutional 1D with no feature selection. And then our improved model included feature selection and kernel size adjustment. And then our final model included residual connections and data augmentation. So this included noise, prevention of overfitting, et cetera. Cool. And then for the final results, we averaged over 3 runs for the purpose of this presentation. So we achieved an accuracy of 94.3%, a precision of 92.7, recall of 95.2, and an F1 score of 93.9. So it might be easier to visualize in the confusion matrix. So we correctly classified 95 tumors and misclassified 6 tumors.
And for analysis, we compare our module performance with logistic regression and SVM. So both of these traditional methods performed well, but our module achieved higher recall and F1 score. And additionally, our module's performance is now only about 3 percentage points away from today's leading modules.
So in terms of limitations in future work, before I get into what's covered on the screen here, as I mentioned earlier, it's almost impossible to get a full picture, especially in clinical settings. We only have data with regards to the size, the shape, and whatever physical features we have of the tumor. But there's features like clinical history for a patient and how many years they may be seeing a clinic that could influence these results. So this is a purely data-driven decision, but defining an accuracy based on solely that may not always provide the full picture, which is a consideration that can come in any clinical setting. So in terms of the data training, the first limitation was the data set was relatively small because it's publicly available in Kaggle, and large-scale intensive data sets used in clinical settings usually are pretty hard to find, so they're not immediately available. Like you mentioned earlier, they might not be able to fully generalize due to diverse patient populations. So for example, if these tumors aren't from a diverse host of patients from different ethnicities, races, etc., they may not be able to be generalized across everyone. And then finally, the numerical features, while informative, do not encompass all the factors that might influence a diagnosis. And in terms of future work and the directions that we would take going forward, we would investigate the integration of domain-specific features, races, ethnicities, and include genomic data to improve prediction accuracy and may provide more accurate results, not necessarily on a number standpoint, but in terms of being able to actually provide insights to people.
Team 5.
Yeah, so our project is basically fine-tuning ESM3 with contrastive preference optimization for antigen-specific antibody design.
Yeah, as we learned in Professor's lecture in protein function optimization, antibodies are a vital part. They are a cornerstone of modern medicine. They are used in various of places like in therapies for cancer, to treat autoimmune diseases, infectious diseases, etc. And they have very high specificity and affinity for target antigen, which makes them unique in therapeutics. But the problem with this is engineering antibodies for optimal performance often involves improving their binding affinity and stability, which requires deep insights into their structural and functional relationship. And so protein language models like ESM3 can be leveraged to predict the structure and the functional properties of antibodies. This can give us a design of antibodies with enhanced biological functions. So the motivation for this is, given the vast amount of foundational models available for modeling protein evolution, it is obviously non-trivial to fine-tune them for specific biological purposes. In particular, ESM, which is trained on millions of years of evolutionary data, it still requires further optimization in case of more variable proteins such as antibodies. So we use ESM as a generative tool to create antibodies with high binding affinity for a particular antigen target. In this study, we are particularly focusing on trastuzumab against HER2. Trastuzumab is a targeted therapy drug that treats certain type of cancer. HER2 is a protein that controls cell growth and division. When some types of cancer cells produce too much HER2, it can cause them to grow and spread much faster. We have used a data set that contains approximately 500,000 trastuzumab CDR variants that are split into high, medium, and low classes based on their binding affinity to HER2. We will utilize ESM3 to generate initial embeddings for the antibody CDR3 sequences. We will then sample two sequences from the data set, one with high binding and another with medium or low binding, and then perform contrastive preference optimization in order to steer ESM3 to predicting sequences with high level of binding. So CPO, or contrastive preference optimization, works by learning from pairs of examples where one is preferred over the other. So this allows the model to emphasize on features associated with higher performances or utility. We'll finally evaluate the space, embedding quality, and sequence generated. So in related work, we have ESM2, which is an encoder-only model designed to process input proteins and extract rich embeddings from downstream tasks. Whereas ESM3, on the other hand, extends its capabilities by incorporating decoder functionalities, enabling tasks like sequence generation and structure prediction, which are crucial for antibody design. Antigen-specific antibody design via direct energy preference optimization. This is another study which leverages structural information to guide antibody design by optimizing energy-based scores for preferred configurations. However, structural data for antibodies is sparse, limiting the approach's scope. Sequence-based methods are often favored due to their relative abundance or sequence information. Antibody or IGLM is another few models which even some people spoke about it in the last presentation. These are antibody-specific proteins language models tailored for analyzing antibody sequences. While they are effective, they struggle to generalize across the broader structural and evolutionary diversity of proteins.
Yeah, so going into like a little more detail of why we chose ESM3 in particular, Basically, unlike specific language, antibody language models such as IGLM and AntiBERT, ESM3 actually incorporates evolutionary information during its pre-training process. And also, unlike ESM2, ESM not only has a built-in sort of decoder capabilities, but also is trained on multiple modalities such as secondary structure as well as solvent accessibility. And for fine-tuning ESM3, what we the motivation behind was that we wanted to prevent interference across the structure and function tracks. So we froze all of those parameters apart from basically the sequence embeddings and the sequence decoder heads, which ended up in 2.5 million tradable parameters. And going into a little more detail of contrastive preference optimization versus direct preference optimization. So contrastive preference optimization can be seen as basically like a general approximation of direct preference optimization. And it's known to be a little more memory and speed efficient, which is why we chose it. And at a high level, it basically trains models to avoid generating adequate but not perfect translations in like translation encoder-decoder tasks. And in terms of the loss function itself, sort of the first term encourages the model to rank like preferable or winning responses higher than the non-preferable ones. And the second term in the loss function basically encourages the model to align with the training data and predict the preferred variables more likely and the non-preferred variables less likely. As for the data that we used, we basically had a data set that included 500,000 trastuzumab CDR3 variants. And they were categorized based on their binding affinity with HER2, either high, medium, or low. And actually, to construct the CPO data set, we need pairs of data points. So we need a high binding affinity sequence and a low binding affinity sequence. So in order to build this out, we kind of followed a similar method that Clean did, where we took the embeddings of all the CDR3 sequences that were generated from ESM3, and we extracted the top 1 million most similar embeddings based on their cosine similarity. And we used that as our training data set. And this resulted in sort of a ratio of 1 positive sequence for 4 to 5 negative sequences.
Yeah. Once we have the fine-tuned model, we want to evaluate if our model is able to improve the richness of the embedding space. Specifically, we wanted to see if our model is able to differentiate the high binding sequence and low binding sequence in the embedding space. So what we did was we calculated 2 cluster centers by averaging all the embeddings of high binders in the training set and also the low binders in the training set. And in the test set, we've classified each sequence as a high binder, low binder by calculating if it is near to the high binding cluster center or the low binding cluster center. And we observed that we compared the evaluation between the base model and the fine-tuned model. And we see that we only have slight improvements in the recall, and all other scores remain mostly the same. And even the AOC curve has only the marginal gains. The problem might be we are mostly classifying very good true positives, but we are having a lot of false positives. And in the next slide, we wanted to see how the newly generated sequence, how close they are to the high binding sequences. So we computed the average edit distance for each generated sequence throughout the all higher binding sequences we had. And we observe 3 models. One is the base ESM3, one is the IGLM, and one is the fine-tuned ESM3. The left graph is the one that denotes that shows the average edit distance. We see that our model performed better than both base model and also the IGLM. And on the right side, we have the plausibility of the sequences. Like we used AntiBERT to compute if our generated sequence is plausible or not. Again, we have compared the difference between base ESM3, IGLM, and fine-tuned model. And we see that our model is doing better than both base model and IGLM. And next, for the structural evaluation, we folded our model using AlphaFold 3, and relaxed the model and computed the interaction energy. And the sequences on the top are the ones that we have selected from a fine-tuned model, and these sequences are we selected the sequence by using antibody to find the top 5 most plausible sequences, and the bottom sequences are the sequences from our training set. And as we can see that we computed the interactive energy using FoldX. We observed that our model, the sequences generated by our model has interactive energy almost as good as the training set representation.
Yeah, so in terms of discussion and future work, I guess what we found was that fine-tuning ESM3 with antibody preference data, despite ESM3's limited exposure to prior antibody data, actually ESM3 was not the ESM3 model that we fine-tuned was not trained on any antibody data at all. It can actually generate plausible and comparably high affinity CDR3 sequences, which is pretty exciting. And one thing that we also noticed was that basically fine-tuning only the sequence track leads to enhanced sequence generation, but still has a limited impact on the overall embeddings, as we found out from basically our classification evaluation. And it also can potentially interfere with other structure and function tracks despite our efforts in freezing those parameters. So in terms of avenues for future work, we'd definitely like to train on more data sets, so include more pairs of positive and negative sequences. We want to basically fine-tune a larger ESM3 model that was maybe pre-trained on actual antibody data. And we also want to integrate structure and function information into the fine-tuning process to maybe add to the richness of the data itself.
Hi everyone. Today we'll be presenting our work on reinforced fine-tuning of protein language models, wherein we propose a new percentile-based approach to kind of ensure diverse protein generation. So just to introduce the problem, so we all know that protein language models actually have shown quite a lot of promise in terms of novel protein sequence generation, as well as the drug discovery applications and then like protein folding prediction tasks. Right now, the way to kind of take these pre-trained models and actually make them work on a particular downstream task is to kind of fine-tune them. A typical approach is supervised fine-tuning, wherein what you do is like you have a ground truth data set of like that based on the downstream task of the set of proteins which you want to kind of fine-tune the model on, and you use that ground truth data set to kind of tune your model. The problem with this approach is you need those protein samples. So it's very difficult to kind of generate that ground truth data set at a larger scale. So another alternative approach is sort of an online fine-tuning, wherein what you do is like you use the generative model, the LLM or the PLM basically, to kind of sample protein sequences, and you use some sort of like scoring on those protein sequences to create your training data set. So this is more like an online mechanism wherein you are using your generative model to create to like sample good protein sequences and using them to kind of then do fine-tuning. So there are essentially 3 components to this framework. So you have the initial generative model, which is like maybe a foundation model, which is represented by theta ref, and that's the initial model you initialize with. Now using that model, you sample certain protein samples. So you basically sample maybe like K samples from that model. The second component is your reward model. So that reward model basically then scores those protein sequences based on certain properties which you want to ensure in those proteins. Once the reward model scores those proteins, there's a filtering mechanism which basically filters the right set of proteins based on whatever condition you define there. So in this case, for instance, this method actually uses a threshold wherein all proteins sequences which are above or which achieve a score of above certain reward threshold are selected. Once these are selected, actually, they are then included into a fine-tuning buffer, and that kind of serves as the ground truth for the model. So the generative model is now fine-tuned using these samples, and is continuously updated, and this cycle keeps on repeating. The problem with such an approach is, although it achieves high reward because you are kind of having you kind of enjoy a reward threshold, it does that at the expense of diversity. So as the training progresses, what we find is like the diversity of the generated protein sequences actually suffers a lot. So another approach which has been proposed is basically instead of so instead of actually using a reward threshold, what you could do is you could just select the best sample out of those k samples which you are basically sampling or generating from the model. Again, I mean, because you are restricting the number of generated samples which you are selecting to be like the best sample basically. So it kind of, what we find is like this approach also kind of leads to generating nearly identical sequences with very minimal diversity, although it again ensures higher reward in terms of like the proteins which are being generated. So essentially both these approaches currently actually suffer from like a lack of diversity in the generated proteins.
So in order to demonstrate some of these limitations empirically, we fine-tuned the MPNN ProGen extra large model, which is trained on the Cath 4.2 dataset. So what this model just does is it takes in a prompt and generates a corresponding amino acid sequence. And our fine-tuning task here is to optimize the reward for some target property that we want to see in the sequences, but also maintain high sequence diversity while we're doing so. So we evaluate this just on the reward, the diversity score, and then KL divergence. So our reward model basically focuses on three main criteria. So first is just the validity of the sequence. So if we're generating a sequence that's either empty or contains characters that don't correspond to any real amino acids, it gets a validity score of 0 and it gets 1 otherwise. And then sort of our main reward is this hydrophilicity balance score. So here we're looking at the ratio between the hydrophobic and the hydrophilic amino acids in the sequence. And we basically just have this optimal ratio of 0.5. So if the sequence achieves this optimal ratio, then it gets a reward score of 1. And then the further away it gets from that, the optimal ratio in either direction, the lower the reward score gets. And then finally, we have the diversity score, which is just the weighted sum of average Hamming distance within each candidate batch, and then the overall uniqueness of the sequence. Now, when this diversity score goes to 0, it means that the model is experiencing diversity collapse, which means that it goes to what we mentioned earlier, which is just generating identical sequences in each case. So the diversity score that we mentioned there is actually not used in every experiment, but we'll note when it is being used. So first here we have the reward graph for Raft. And what we can see here is actually that Raft is really good at helping us achieve optimal rewards. So basically every candidate count setting is achieving optimal rewards except for 2 of the lower count settings, which are converging between 0.8 and 0.9. But then when we go to actually the diversity graph of Raft, we're seeing sort of what this key issue is, which is that while we're achieving optimal rewards, we're actually converging to diversity collapse in almost every setting. So actually only one candidate count setting maintains some diversity throughout the training progress, and that is C equals 16. But actually, if you'll remember from the last graph, C equals 16 is actually one of the settings that achieved suboptimal results. So what we're seeing here is we can sort of either achieve optimal rewards or maintain high diversity, but it seems to be hard to do both at the same time. So then here we have Raft plus that diversity term that we were talking about earlier. So what we see here is on the right graph is that actually diversity is looking a lot better than it did in the Raft without the diversity addition. But actually, at the same time, our reward graph is looking significantly worse. So actually, the only setting of diversity weight that is achieving optimal results here is the diversity weight with zero weight on the diversity term. So actually here we're seeing this trade-off between the reward optimization and maintaining high diversity, and seeing that it's very difficult to do both at the same time. And then finally here we have the rewards from REFT, which is the other fine-tuning training algorithm that we talked about. And here we're seeing very similar results. So actually the few reward thresholds that achieve optimal rewards on the left side graph are actually end up being the same ones that achieve, or that converge to diversity collapse on the right side graph. So we're seeing the same sort of trade-off between optimizing rewards and maintaining high diversity.
Yeah. Based on all the experimental results of existing method or existing method with some diversity regularization, we can see that some challenges exist in current methods while fine-tuning the PLM. So firstly, we see clearly diversity collapse of the current existing method at the REFT, RIFT, or other methods. And another thing is that even if we add some diversity regularization into our current method, we find that they are hard to fine-tune the hyperparameters, or hard to trade off the reward and diversity. And in fact, some existing method just propose to track this method, track this problem by adding some care regularization, diversity regularization, but this will induce a multi-objective optimization. Is very hard to achieve. And to handle this problem, our key contribution of this work is to propose a novel percentile-based reward filtering mechanism based on, it's also a reward filtering mechanism that naturally balances the diversity and performance rather than use a fixed threshold such as we see in RIFT. Or just use top K, best of K sampling. We adaptively select a sequence based on their relative performance. And besides, we achieve, if we can, and the pipeline is quite simple, just focus on this part. The other part is quite similar. And we just replace the sampling part, and we replace the best of K or with a percentile-based sampling just to. And looking at our experimental result, we can see that our method can effectively balance the reward optimization and the diversity. And you can see the conversion points. This is the conversion points. Most of our most of the conversion point of our method converge to a very high, optimal reward at 1, and while maintain a high diversity.
It's been a little over 10 minutes, so try to wrap up in the next minute.
Yeah. And this is just what we want to put, and just make a quick conclusion. And we just show how to adapt the techniques of RHF to fine-tune PLMs, while existing methods like REFT struggle to balance the reward and diversity. And in this paper, we just want to find a general way to to balance this, to address this limitation without any regularization or human collected data. And our work not only solve these limitations and just want to open a new existing possibility for diverse protein generation. And yeah.
Hi everyone, we're Teammate, and our project is extending DNA sequence modeling performance of nucleotide transformer. This is our agenda. And so for motivation, the nucleotide transformer is a foundation model for DNA sequence with strong performance on various downstream tasks like enhancer prediction and promoter detection and splice site identification. And so it is pre-trained on a large data set of 850 species using max language modeling with 6-mer tokenization, and it captures like important genomic elements. So the diagram on the right shows how it generates embeddings from DNA sequences, which are then used by models to make predictions. So, but however, there's still potential to further enhance the effectiveness of that. So in this project, we focus on exploring strategies to improve and optimize its capability. So we further explore its performance under different settings. So first, we compare k-mers and byte pair encodings and evaluate byte pair encodings potential for improving computational efficiency and sequence classification, the downstream task performance of it. And for fine-tuning tasks, we explore path methods to adapt nucleotide transformer for downstream DNA sequence classification tasks. And lastly, we explore the mixture of LoRa experts to improve generalization across tasks by using multiple LoRa experts. So a brief introduction about nucleotide transformer is that it is built using an encoder-only transformers and using 6-mer tokenization with rotary embeddings. The model comes in scales, including like verse 1 with up to 2.5 billion parameters, and verse 2 with small versions from 50 million to 500 million parameters. And it is trained using mass language modeling on datasets with 300 billion to 1 trillion tokens. So for fine-tuning, it uses techniques like IA3 to adapt nucleotide transformer for downstream tasks. So the graph on the right is the performance from the paper. It is used across tasks such as chromatin profiling, regulatory elements, and also splicing. So the right for a column of each plot is the performance of it, and it shows improvements. So in this project, we focus on nucleotide transformer verse 2 with 50 million parameters due to computational constraints, and we test its performance on 18 downstream tasks. So for tokenization on the left, we see k-mer tokenization, which splits sequences into fixed length substrings, and it captures local sequence motifs and context, but there's also a limitation of it, as the fixed k-mer length can restrict flexibility and also the computational efficiency. And so on the right, there's byte pair encoding, which offers a more dynamic approach by iteratively merging frequent token pairs and captures recurring patterns and motifs across sequences from datasets, but it reduces the sequence length and vocabulary size, but also risks like losing information when merging them.
The parameter efficient fine-tuning, or PEFT, is an efficient essential technique for adapting the large models like the nucleotide transformer to downstream tasks without incurring excessive computational costs. So one prominent PEFT method is LoRa, or low-rank adaptation. LoRa fine-tunes only small rank decomposed matrices, making it computationally efficient and ideal for resource